Good morning. Myself, Dr. Anil Kumar Chaudhary, in charge Department of Genetics and Biotechnology, College of Agriculture Science, Tirthankar University, Moradabad. Today we are discussing on the paper crop improvement second learning crop immunization. So the topic is germplasm unit third. So when we talking about the germplasm, so a lot of questions in our mind. What is germplasm? So today we discuss about its difference and its use, a lot of things about the germplasm. So firstly, I tell you about the germplasm. Germplasm are the genetic resource. It means it is the total genetic material such as seed, like seed, maybe tissues, and DNA sequence that are the maintained for the purpose of animal and plant conservation efforts, agriculture, and other research use. These resource may be taken in the form of seed collection, storage, seed bank. Lot of things and lot of candidates about the NBPCR, National Bureau of Plant Genetic Research, the headquarters near ICR campus, Sunny Valley. Lot of germplasm of the plants, roots, formatting plant, seed format, root format, tissue, different formats stored in seed banks, DNA banks, and a lot of trees growing in nurseries, animal breeding lines maintained in the animal breeding programs, tree banks. Germplasm collection can be ranged from the collection of wild species, right, domesticated breeding lines that have the undergone extensive human collections, selections. Germplasm collection is the important for the maintaining of biological diversity, the security of the food. Because a lot of germplasm have the different type of the quality, different type of the characteristics. Like some wild germplasm, mustard I know personally, and grow the different germplasm in Brassica juncea, having the best quality against the disease, against the insect resistance. So that is very essential for the security in future. Now we are discussing about the lentils. Lentils. You can say the very old variety, primitive variety, grown in earlier times of a particular region. That is very famous sweets. Lot of farmers grow at the present time for because this variety is very useful. This variety type seeds is very tasty cereal, wheat, bajra, very nicely. And second is cultivars. So two types of the cultivars, obsolete cultivars, that is the very old cultivars covered the lot of area. And the third one is the modern cultivar. It means presently covered a lot of area, presently showing the variety largest area, like for example HD296 variety of wheat crop. This variety covered the lot of area in India. That is the very famous HD2967 variety, the very famous variety of wheat crop. We can say this variety modern cultivars. And the fourth one is the advanced breeding lines. When we develop the different type of the genotypes and lines under the pipe and develop the new genotypes for enhancing the breeding programs. So that is very essential and very useful lines in breeding program and developing a new lines or variety. And number five, wild from the cultivated species. That is the wild species, and change the wild from cultivated things. And number six is wild relatives. That is wild relative. It means, for example, in Brassica, 5-7 is the cultivated species, and Other 20-30 species is wild, like Seneca salva. So we can take the different character against to disease or taste from the wild relatives. That is very useful. Number 7, mutants. In the nature, sometimes lot of species, lot of variety naturally change due to the ultraviolet rays and other problems. So that is the type of germplasm. And next one is land races. They are nothing but primitive cultivars were being selected and cultivated by the farmer for many generations without systematic plant breeding efforts. In this slide we easily explain about the land races, but now I tell you about the properly land races. The main drawback of the land races, they are less under and low yielders. ये उनकी उपज जो है बहुत कम होती है. इट मीन्स जो आज वैरायटियां हैं और जो पुरानी वैरायटी हैं उन दोनों में अगर डिफरेंस किया जाए तो लैंड रेसेस की यील्ड तो बहुत कम होती है, लेकिन क्वालिटी कंपैरिजन बहुत अच्छी है, बेस्ट कंपेयर टू हाइब्रिड एंड करेंटली जो प्रचलन में वैरायटी है लैंड रेसेस. Second, affiliate cultivars, they are the varieties developed by the systematic breeding efforts which are popular earlier and now have been replaced by the new variety. नई वैरायटी आती है 5 साल 10 साल धूम मचाती है, तो उनको रिप्लेस करने के लिए वैरायटी आ जाती है. Improved variety of recent past are known as the affiliate cultivars. Affiliate cultivars have several desirable characters. They continued an important part of gene. Next, modern cultivars. About the modern cultivars. Every a lot of variety is by different institutions, ICR universities and ICR research centers. And advanced breeding line, they are the pre-released plant which have been developed by the plant breeders. Modern scientific breeding program, they are known as the advanced lines, culture line stocks. These lines are not yet ready to release to them. They often contain valuable gene combinations. Next is wild from the cultivated species. While from the cultivated species, they are available in many crop plants. Such plants have generally high degree of the resistance to biotic and abiotic stress and unutilized. They can easily cross with the cultivated species, which wild forms of many crop species are. Number 6, wild relatives. This slide I explained about the wild relatives. Those naturally occurring plant species which have common crop can cross the crop species referred to the wild relatives or wild species. The wild relatives include all other species which are related to the crop species by the descent during their evolution. Both these groups are sources of valuable gene for biotic and abiotic stress of quality traits. Mutation breeding, that is the very famous and popular part of breeding. Everybody know about the mutation breeding, but before the explanation of the mutation breeding, mutants, it means was changed through the naturally, it means spontaneous or induced. So mutation breeding is used when the desirable character is not found in the genetic stocks of the cultivated species and their wild relatives. Mutations do occur in nature as well as can be induced through the use of physical and chemical mutagens, which are change to a genome sequence or other things through the physical method and chemical methods. In physical methods, a lot of rays, X-rays, gamma rays, alpha rays, beta rays, and in chemicals, so lot of chemicals we can use through to change the DNA sequence and DNA. So thank you.